Have you ever wondered what makes you, you? What is it that defines your unique characteristics, your traits, and even your potential? It's a question that has fascinated scientists, philosophers, and curious minds for centuries. Deep inside almost every cell in your body is a very special molecule called DNA. This molecule is incredibly important because it carries the genetic instructions used in the growth, development, functioning, and reproduction of all known living organisms and many viruses. DNA is like a blueprint. It holds all the instructions for building and running a living thing. Imagine it as a detailed manual that tells your body how to function, how to grow, and even how to repair itself. Every tiny part of you, from the color of your eyes to the way your heart beats, is encoded in this remarkable molecule. It's what makes you unique. Today we're going on a scientific adventure. We're going to dive deep into the world of DNA and uncover its secrets. This journey will not only be educational, but also incredibly fun and engaging. We're going to extract DNA from a strawberry and see it with our own eyes. This simple experiment will show you just how accessible and fascinating science can be. You don't need a fancy lab or expensive equipment, just a few household items and a bit of curiosity. DNA is found in every living organism. Plants have DNA, animals have DNA. This universal presence of DNA is what connects all life on Earth. It's a common thread that links us to every other living thing, from the tiniest bacterium to the largest whale. Even microscopic bacteria have DNA. These tiny organisms, invisible to the naked eye, carry the same fundamental building blocks of life. It's amazing to think that something so small can hold such complex information. It's the secret code of life. This double helix structure, with its twisting ladder-like appearance, is the key to understanding how life operates. Each rung of the ladder is made up of pairs of nucleotides, which are the basic units of DNA. These sequences determine everything about an organism. And guess what? We can unlock this secret using a simple, fun experiment you can do at home. This hands-on activity is perfect for families, students, or anyone with a passion for discovery. It's a great way to bring science to life and see the wonders of DNA firsthand. This experiment lets us see DNA in action. By extracting DNA from a strawberry, we can observe the physical substance that carries genetic information. It's a tangible way to connect with the abstract concepts of genetics and molecular biology. It's a chance to explore the microscopic world. When we look at DNA, we're peering into the very essence of life itself. This microscopic world is full of wonders and mysteries waiting to be discovered. Get ready to be amazed as we unveil the building blocks of life. This journey into the world of DNA will open your eyes to the incredible complexity and beauty of the natural world. So let's embark on this adventure together and unlock the secrets of life. Why strawberries, you ask? Strawberries are like little DNA factories. They have a lot of DNA packed inside. This makes it easier for us to extract and see it. Strawberries have another secret weapon, enzymes. These enzymes help break down the tough cell walls that protect the DNA. Think of it like this. The enzymes are like tiny scissors, snipping away the walls to release the DNA treasure inside. Plus, strawberries are soft and easy to crush. This makes them perfect for our experiment. We can easily break them down and release the DNA without any fancy lab equipment. Before we begin our DNA extraction, let's gather our scientific tools. This is an exciting journey into the world of genetics, and we can do it all from the comfort of our home. Don't worry, you won't need a fancy lab. We're using everyday items found right in your kitchen. You will need a few simple things that you probably already have at home. Fresh strawberries, the stars of our experiment. These juicy fruits are packed with DNA, making them perfect for our extraction process. Make sure they are ripe and fresh for the best results. Plastic bag, our DNA extraction chamber. This is where the magic happens. We'll use it to mash the strawberries and break down the cell walls to release the DNA. Sieve to separate the solids from the liquid. This will help us filter out the larger pieces of strawberry, leaving us with a liquid that contains the DNA. Cup to collect our precious DNA extract. A clear glass cup works best so you can see the results of your hard work. Table salt. This helps the DNA clump together. Just a pinch will do the trick. Salt neutralizes the charge on the DNA molecules, allowing them to stick together. Liquid dish detergent breaks down cell membranes to release DNA. The detergent dissolves the lipids and proteins that make up the cell membrane, freeing the DNA into the solution, cold ethyl alcohol. This helps the DNA become visible. Make sure it's very cold, as this will help the DNA precipitate out of the solution. You can store it in the freezer before starting the experiment. Teaspoon for measuring our ingredients. Precision is key in science, so make sure to measure accurately. Wooden stick, our DNA scooper. This will help us collect the DNA strands once they become visible. A wooden stick works well because it won't react with the chemicals we're using. Now that we have our equipment, let's put on our scientist hats and begin our DNA extraction adventure. Get ready to dive into the fascinating world of genetics and see the building blocks of life with your own eyes. This hands-on experiment is not only educational, but also a lot of fun. So gather your materials, follow along, and let's make some scientific discoveries together. Our first step is to release the DNA from its protective home inside the strawberry cells. How do we do that? By crushing those cells. Place your strawberries inside the plastic bag. Make sure it's sealed tight. Now, it's time to get our hands dirty. Squeeze, mash, and crush those strawberries until they turn into a smooth paste. The more you crush, the more DNA we'll be able to extract. We're breaking down the cell walls and releasing the DNA into the mix. It's like popping open a piñata filled with DNA goodies. Section 5, Step 2, breaking down the cell walls. Remember those enzymes we talked about? Well, it's their time to shine. Enzymes are like tiny molecular machines that speed up chemical reactions. In this case, they help us break down the cell walls of the strawberries, making it easier to extract the DNA. We're going to add a special mixture to our strawberry paste that will help break down the cell walls even further. This mixture includes common household items that you might already have in your kitchen. Carefully add a teaspoon of salt and a teaspoon of dish detergent to the bag. The salt and detergent play crucial roles in this process. The salt helps the DNA clump together, making it easier to see and extract later on. Meanwhile, the dish detergent breaks down the cell membranes, which are made of lipids and proteins. These membranes are like the walls of a house, protecting everything inside. By breaking them down, we can access the DNA within the cells. We need to break them down to reach the DNA inside. Think of it as demolishing a building to get to a hidden treasure. Seal the bag tightly and gently massage the mixture. This helps the salt and detergent mix thoroughly with the strawberry paste, ensuring that the cell walls are broken down effectively. Avoid creating too many bubbles. Bubbles can interfere with the process and make it harder to extract the DNA later on. We want to be gentle with our DNA. The goal is to keep the DNA strands intact so that we can observe them clearly. By taking our time and being careful, we can ensure the best results for our experiment. This step is crucial in our journey to understand the building blocks of life. By breaking down the cell walls, we are one step closer to isolating and studying the DNA, which holds the genetic blueprint of the strawberries. DNA extraction is a fascinating process that allows scientists to study the genetic material of organisms. It has applications in fields ranging from medicine to agriculture and even forensic science. As we continue with our experiment, remember to take detailed notes and observe the changes carefully. Each step brings us closer to unlocking the secrets hidden within the cells. The more we understand about the cellular structure and the role of DNA, the better equipped we are to make advancements in science and technology. So let's proceed with curiosity and precision. Section 6, Step 3, filtering out the larger pieces. Now that we've broken down the cell walls, it's time to separate the DNA from the rest of the strawberry goop. We'll use our sieve and cup for this step. Place the sieve over the cup. Carefully pour the strawberry mixture into the sieve. The liquid will pass through, leaving behind the larger pieces of strawberry. The liquid we've collected contains our precious DNA. Section 7, Step 4, precipitating the DNA. Get ready for the grand finale. We're about to force the DNA out of hiding. This is where the cold ethyl alcohol comes in. Tilt the cup slightly, very slowly pour the cold ethyl alcohol down the side of the cup. Don't mix or stir. The alcohol should form a separate layer on top of the strawberry liquid. Watch closely. You should see white, cloudy strands forming in the alcohol layer. That's the DNA. Section 8. The science behind the magic understanding DNA extraction. What we just witnessed is pure scientific magic. Let's break down what happened in each step. Crushing the strawberries broke open the cells, releasing their contents. The salt helped to clump the DNA together, making it easier to see. The dish detergent dissolved the cell and nuclear membranes, which are made of fats and proteins. This freed the DNA from within the cell. Finally, DNA is not soluble in alcohol. So, when we added the cold alcohol, it forced the DNA to precipitate out of the solution. That's why we saw those white, stringy clumps. Section 9, Conclusion, a journey into the microscopic world. Congratulations, scientist. You've successfully extracted DNA from a strawberry. You've seen with your own eyes the molecule that holds the blueprint of life. This experiment is a fantastic way to learn about DNA and cell structure. It shows us that science can be fun and exciting. Now that you've unlocked this scientific secret, keep exploring. Try extracting DNA from other fruits like bananas or kiwi. Share your discoveries with your friends and family. Who knows what other scientific wonders you'll uncover?